What if the genesis of life, the very spark that ignited existence as we know it, could be replicated within the confines of a laboratory? A compelling question indeed. Let's journey back to the primordial era, roughly 4 billion years ago, long before dinosaurs roamed the earth and bacteria thrived. Picture a world teeming with the potential of life, but life as we understand it hadn't yet taken form. The primordial soup of the early earth was just that, a possibility. It was within this realm of potential that a molecule called RNA, ribonucleic acid, made a monumental leap. It replicated itself. This self-replication set off a domino effect of sorts. The copies made more copies, and, over millions of years, RNA gave birth to DNA and proteins. These components came together to form a cell, the smallest unit of life able to survive independently. Fast forward to the present day, where scientists at the Salk Institute for Biological Studies in La Jolla, California, have made a significant stride in supporting this RNA world theory. They have crafted an RNA molecule within test tubes that can accurately replicate a different type of RNA. This achievement brings us one step closer to the grand goal of growing an RNA molecule that can accurately replicate itself. Imagine the implications of such an achievement. As Gerald Joyce, president of Salk and one of the authors of the study, puts it, then it would be alive. This is the path to understanding how life can arise in a lab or, in principle, anywhere in the universe. While there is still a considerable distance to cover before we can definitively say this is how life on Earth began, the scenario tested likely mirrors one of the earliest stages of evolution. This concept, as described by English naturalist Charles Darwin over a century and a half ago, is a stepping stone toward understanding life's evolution. The scientists overcame a significant hurdle to the plausibility of the RNA world theory. Until this point, no RNA molecule in a lab could create copies of other RNA that were both sufficiently accurate and functional. An RNA molecule must make copies that closely mirror the original to achieve the delicate equilibrium that governs Darwinian evolution in nature. If the copies deviate too much, the RNA's abilities deteriorate and the consequences are rapid and severe. However, the copying can't be flawless all the time. Without room for errors, the RNA would be unable to adapt when its environment changes, a necessary quality for living creatures in the wild. The experiment's findings suggest a delicate balance between accuracy and adaptability is necessary for the evolution of life. The replication of RNA in a lab setting is a monumental stride in our understanding of life's origins. This breakthrough not only helps us comprehend how life may have arisen on Earth, but it also opens up possibilities for creating life in a lab. The implications of this research are profound, challenging our understanding of life and potentially guiding us toward the genesis of life in a laboratory.